Welcome everyone. Thank you for joining us for today's webinar. My name is Dr. Kumar Jitendra and I am the Chief Editor for Microbiology India. Microbiology India is a leading publication for biopharmaceutical industry. So today we are presenting the power of precise PK data in generic drug development, which is in association with Inhalation Sciences and Apex Chromatography. So just little housekeeping before we get started. If you have any questions during the presentation, please type them into the question box in your control panel and I will bring them up during the presentation, and we will also have time for questions at the end. And one very important thing, parts to all attendees join as a co-organizer here. Please don't push that red button showing below your screen, otherwise session will end immediately. So now, without further ado, we will turn the time over to our session. Our presenter today is Paolo Raffeli. Paolo has over 20 years executive experience in the life science and medical device industry with extensive sales and marketing.
Thank you, Kumar. This is Paolo Raffeli. I'm the Chief Commercial Officer of Inhalation Sciences. Inhalation Sciences is a Swedish company based in Stockholm, and we specialized in lab instrumentation for inhalation drug R&D. Now, today you see here the panel of speakers. My colleague Mikael Mikko, working here as Product and Sales Manager. We do part of the presentation. Per Gerde, the Chief Scientific Officer and founder of the company, will be available later on for the Q&A session. Maria Malmlöf is the in vitro director, so she is our specialist for dissolution tests. So she will present and she will be in the lab live demos. And also Beatrice Biering, she's the product engineer. So she's working with technical services. She will present the PreciseInhale instrument in the lab. So here is the agenda for today. After a short introduction on the dissolution test and bioequivalence pathway, Mikael will present the portfolio, what we can offer in terms of products and services, and then we'll present the PreciseInhale aerosol generator, which has some unique features, and Beatrice will show you live in the lab. Then we'll go on with the Solvit, which is really the key instrument in the dissolution absorption test, which is, we will see, an important part of pharmacokinetic testing for generics drug development. And then in the end, we'll have a Q&A session. Now, you see, this slide, introduction slide, wants to summarize the overall issue with dissolution testing for inhaled drug products. So this is a quotation you see here and down here, the quotation marks, from an article that came out recently this year. And this article reports a complete PK study of a lung cancer drug in development, which was planned to be delivered as an inhaled drug. There is a paragraph in which the authors of this article comment the dissolution tests they performed. And you see here, I just took some excerpts and I highlighted in bold some words. But I thought this is an excellent summary of all the problems there are today with dissolution testing of inhaled drug products. So, there are no standardized method or guidelines describing in vitro dissolution tests for inhaled products. There is a difficulty reproducing physiological conditions, what happens in the lungs, with in vitro dissolution methods available today. They had also issues with particle agglomeration between the API and the excipients, so that's something that often happens when working on dissolution tests. Then there is another group of comments within the same paragraph. So they were recommended to interpret the dissolution profiles with caution because of the quality of the data they could generate. And overall, their comment was that the methods used today, they are just inadequate to what they would like to achieve in terms of in vitro, in vivo correlation as well, which was judged as poor. So they were calling in the end for further development and, highly awaited kind of, developments in this area to solve these drawbacks. So what we want to do today is we want to present our system, our solution to all these issues, basically. So it's a relatively new system. It's not very well known, but we want to share this with you, and we'll also present already published data. So it's a new system, but we have solid data that we will partly present today. And if there is interest, of course, we can organize more webinars in the future and follow up in deeper details. So if we speak about the FDA approach to bioequivalence kind of realization, you see on the right-hand side, you see the standard, let's say the established approach from the agency, and it requires in vitro similarity, systemic PK data, and pharmacodynamic data, so clinical trials. Now, the agency recognizes the difficulty of creating, working with inhaled drug products. So there is a kind of, emergence of novel BE approaches. And there is a set of data that are clearly important and will be more important in the future to establish these novel bioequivalence pathway recognition methods. Now, in particular, in the different, in the set of different data that will be required, we can see this solution has a central position. And again, today, we will present our system to deliver this solution data in a very precise and physiological way already from the in vitro phase. Now I'll pass it over to my colleague Mikael, and he will present our overall portfolio.
Yes, so with the PreciseInhale system platform, we offer flexibility, accuracy, and precision in generating PK data across in vitro and in vivo. And we like to see our platform as an inhalation switchboard, a plug-and-play device. And what you see here in the middle is the PreciseInhale aerosol generator. And as inputs, we can choose different aerosol sources. We can work with dry powder as it is, APIs. Sometimes it's better to work, easier to work with solutions. Then we have an option for nebulizer. And you can also connect any type of inhaler, both DPIs and PMDIs. So it's very flexible on the input, but it's also flexible on the output. So on the output, we have both in vitro and in vivo modules. If you start looking at the in vivo modules, we have an aerosol characterization kit to characterize the aerosol. We use then a Marple cascade impactor, providing MMAD and geometric standard deviation. We also have a cell culture exposure module, ExposeAli. Here you can expose cells in an air-liquid interface, providing permeability assessment or tox testing. But today, we will focus on dissolve it. And this is our unique dissolution and absorption lung testing. It offers physiology-like conditions with a single pass perfusate, generating Cmax and Tmax data. So we'll look more into this one, but we also have in vivo modules, nose-only rodent exposure and intratracheal rodent exposure. And in both these modules, we work with one animal at a time, and that provides us full control or monitoring of breathing parameters and delivered aerosol, generating precision dosing. Intratracheal exposure module is also unique because we bypass the nose and expose the lungs directly. Since a lot of substances normally get stuck in a rodent's nose, we avoid this and get a more clinical-like exposure. And just one more thing with this thing is that thanks to the fact that we use the same aerosol across the different in vitro modules, we can achieve relevant IVIVC in vivo in vitro correlations. All right.
Yeah, thank you, Mikael. This is really what you can see. This is my favorite slide to represent our portfolio. And as Mikael said, you know, the consistency of approach in vitro and in vivo, we think is very important to obtain data that can help you de-risk your clinical trials. So getting solid, reliable data already from the beginning, from the in vitro phases of your research. Now you can see here the PreciseInhale can be used to generate across aerosol across the whole preclinical workflow. And then if you see vertically here with the cascade impactor, you can do the particle size determination. With the dissolve it, as Mikael mentioned, you can generate already Cmax, Tmax kind of PK curves already in vitro. And Maria will describe this system in more details in a moment. With expose Ali, we can expose cells, as Mikael described, with the aerosol. And then with the in vivo modules, we can generate pharmacokinetic data and get systemic blood levels. So this is just gives you a different perspective on our portfolio. And I just want to mention also that we offer services, research services. So all the information that our instruments can generate can be obtained in our lab in Stockholm. And we have had in time, long time cooperations with major pharmaceutical companies and research institutes in providing these services. So from aerosol characterization to in vitro dissolution data, in vivo, as Mikael described. And as a service, we also offer isolated perfused lung, which can be very important to get very specific data on the lungs to give you an answer how long your substance is in the lungs, how quickly it goes through the lungs and ends up in the systemic circulation in a very, very specific way. So at this point, we move on and we will talk a little bit about the aerosol generator PreciseInhale, always from the point of view of generating and depositing the aerosol for the successive dissolution and absorption tests. So it's back to you, Mikael.
Thank you. So we're going to describe how we work with PreciseInhale and how we generate the aerosol. And what you see now is a schematic overview of the PreciseInhale for dry powder aerosol generation. And sorry, you see the system from the side here. We normally start by loading our powder chamber, about 1 milligram per loading. Then we take this powder chamber, put it in place on the instrument, seal it in place, and then we want to aerosolize it. To aerosolize the powder, we use high pressure that can be regulated from 10 to 160 bar, depending on the properties of the powder. So when we activate the software and press expose, the high pressure will rush into this powder chamber, force the powder into this holding chamber. Before it enters that, it passes a nozzle, generating a deagglomerated aerosol, a fine particulate aerosol. To bring the aerosol out, we use a vacuum pump that can be regulated according to your needs. In this case, you have an end filter holder connected here with receiving all aerosol, but it's on this side you can connect in vivo modules or expose Ali. The incoming air is humidified to decrease static electricity effects. And before the aerosol leaves the instrument, we measure the aerosol concentration. This, in combination with the incoming air that we measure the flow rate And if we work with in vivo, we also monitor the breathing parameters of the animal, because this is a sealed system. So the pneumotach up here will register the flow rate and breathing parameters, and that gives us precision dosing. One important feature for this system is that we can work with very small amounts of substance. 50 to 100 milligrams is usually enough for a complete pharmacokinetic testing. So this is about the aerosol generation with the high pressure that we use, and the aerosol generated here, illustrated as an aerosol concentration curve. So again, precision dosing is achieved by active monitoring of aerosol concentration and breathing parameters. This is the setup for dry powder aerosol generation. When we work with inhalers, we have a different solution that Beatrice will describe in the lab. Then we don't use the high pressure, and the same with the nebulizer. But the idea with the precision dosing is the same, that we measure the aerosol concentration and flow rates. So we can offer to de-risk clinical trials with accurate PK in vivo profiles. So PreciseInhale generates accurate PK data at an early stage. Commonly, or alternative aerosol generation tools that are often used in labs are pen-century-like systems. The problem with that is that you get an inaccurate delivered dose. We force the aerosol into the animal, and it's really operator dependent. Another option is the inhalation towers. These are more designed for toxicology studies, where you expose many animals at the same time. However, you always go through the nose only, which gives you a higher gastrointestinal exposure at the same time. With the PreciseInhale, we offer ideal aerosol generation, the agglomeration of the aerosol. We provide the precision dosing, and that gives us relevant PK data. So we can deliver the substance into the deep lungs. We have minimal gastrointestinal exposure, and we get curves more illustrating what will happen in the clinic. And this was shown in an article where pen-century was compared to PreciseInhale head-to-head. And it was shown that the majority of the aerosol with that system delivered the aerosol to the trachea region, and thus the gastrointestinal exposure as a consequence. That also skewed up the PK data. So with this system, you get PK data from lung exposure. So let's see here. Before we leave and go to the lab, I'm just showing you how the PreciseInhale is set up for the dissolution and absorption testing with the solvents. For that, we use the PreciseInhale with this holding chamber. In the bottom, we have room for cover glasses, which we deposit the aerosol upon. These glasses are then brought to the solvents, where it's sandwiched together in this artificial lung setup connected to a fraction collector. So with that, I will hand over to Beatrice, and she will display the PreciseInhale inside the lab.
Thank you, Mikael. So Beatrice will connect now from the lab. I will stop sharing the screen here. Yeah. And then there you are.
Yes.
Yeah,
thank
we can
you.
see you. Yeah, good.
Good. Yes, so my name is Beatrice, and I will show you the PreciseInhale setup with the dissolving chamber and also with the inhaler generation. So this is the holding chamber for dissolvate. And behind this plate, we have a pinch valve that either directs the air in exposure flow or in a bypass flow. here, this one is an overflow protection filter, which protects us from potential overflow of the particles. Behind this, we have a blue Nemotac. This is a Nemotac that is measuring the flow, the inhalation flow. And then we also have a Nemotac over here, and this is a Nemotac that is measuring the exposure flow. So if we move on here, you can see that we have another part here, and this is called the critical orifice. And this orifice is used to regulate the inhalation flow. And this inhalation flow can be regulated with these plugs. So you can regulate it from 0 to 116 liters per minute by removing or inserting these plugs. And usually you want to create a pressure drop in your system that is around 4 kilopascal, since most DPIs are made for that. And then you regulate the flow in order to achieve that pressure drop. And this vacuum pressure and flow is created by this vacuum pump that we have over here. And then we also have this Nemotac, which is measuring the exposure flow. And this exposure flow is created from a vacuum pump that is inserted in the stand from the back. And this vacuum flow you can regulate with a knob on the stand. Usually we have this around 400, but you can regulate up to 2000 or more. And we also have the humidifier connected to this exposure flow Nemotac. Yes. And here you can see the Casella instrument that is measuring the particles that is passing through. And this is the end filter holder, which you can have an end filter in. Yes. And then I will also show you this part. So this is the inhaler inductor tube. So you can see that it's a 90 degrees angle. And here you have, underneath, you have a lactose trap. So this lactose trap is filled with water. And when you fire this DPI off, some lactose will fall down in this water trap and will be dissolved. And here on this inductor tube, you also have an adapter, which looks like this. And this is very good because this has the same dimensions as an NGI has for these mouthpiece adapters. And these ones are the similar as you use for NGI. So it fits on here. And what you do is that you put this adapter onto this mouthpiece adapter onto this tube adapter, and you put your DPI inside the mouthpiece adapter. So what's happening in the system is that we pull aerosol up in the chamber, and then we put on our exposure flow, and we will pull this aerosol out down to the filter, the end filter. And another option is to use a PMDI. And this is the PMDI trigger. You put your PMDI here, and this has an automatic trigger function, which will be synchronized within the PreciseInhale, and you will trigger the PMDI, and it will be pushed out and dragged out in this holding chamber. And for the solder setup, inside this chamber, we have a glass cover slip holder. Looks like this. And in these holes, you will put your glass cover slips that will be used with the dissolved instrument. One of these slips you can, for example, also use for SEM imaging. Everything is also steered from the computer as well. And then, as an option, as Micke spoke about, another option is to use powder exposures. And this is the powder chamber mantle that can be exchanged with this inhaler inductor tube. So what you will do is to put this chamber onto this high pressure tube. And here you can see that you have a nozzle, and this is the nozzle where the powder will be shoot out from. And then in this, there's also the powder chamber where you will load your powder. And I will also show you this. This is another option if you're not using Dissolvit. You can use this 300 milliliter holding chamber. We also have a bigger one with the 1500 milliliters, depending on what you're going to close. So I think that's basically it. So thank you very much, and let's go back to the presentations.
Yeah, thank you, Beatrice. And now we will continue with showing some slides about Dissolvit. So the whole idea with Dissolvit is to try to simulate the physiology of the airways. And here is a figure of the airways where you inhale particles, the red dots. And what happens in the airways is then that the particles deposit on the mucus layer in the airways, and the particles are dissolved and absorbed via the lung tissue to the bloodstream, where it is distributed via the systemic circulation. And as I said, the idea with Dissolvit was to simulate the physiology of the airways compared to other methods that may not do that in the same way. So here you can find the same color coding as to the left. So as Beatrice showed, we deposit particles on glass cover slips, red dots on this blue surface, with the particles pointing upwards. And the experiment is run on a microscope, so you can visually see the disappearance of particles when they dissolve. And I will show you this setup in the lab soon too. And when the experiment starts, the particles get in contact with our prepared mucus simulant. And the particles are dissolved into the mucus simulant and absorbed into the perfusate via a membrane. And this perfusate is pumped, and also it should simulate the blood. So it contains, it's a phosphate buffer that is, the pH is set to 7.4, and it also contains albumin for a protein content. And in this, the experiment runs, and you use a fraction collector that samples or collects your sample over time, and you can freely program this fraction collector to collect samples at what time points you want. So this is the way how we try to simulate the physiology of the airways with Dissolvit. And here is just a comparison in between dissolve it and some other more convenient or more used methods used today. And other methods might use then an NDI, for example, to deposit the material. And we use PreciseInhale together with this holding chamber. And we deposit the material on glass cover slips. And in the middle, you see here an evenly deposited aerosol. And that is also you could compare that instead to an NDI deposition where you have those nozzle patterns, which will influence the dissolution of the particles. We have shown that in some publication. Another advantage or difference for dissolve it is that you could use, as Michael has said, both dry powder and inhalers, while you use only inhalers in the NDI. And also the inhalers, the dry powder could then be used for really early on in preclinical studies when you have not formulated it into an inhaler yet. Then another difference is that while doing the dissolution test, you would end up with the profiles, concentration profiles looking like this, with Cmax and Tmax. Doing dissolution testing in paddle over disk or transwell, you will instead have this type of cumulative graphs over time. And to get out some kind of pharmaco-like profile, you need to do some modeling, but instead with dissolve it, you get that directly from the test. And here is the workflow of a typical dissolve it experiment or study. First, you can use PreciseInhale to do some characterization of the aerosol used, using Marple cascade impactor to determine the aerodynamic size of the particles, which of course could influence the dissolution of the particles. Then, as said, you use PreciseInhale again to deposit aerosol on glass cover slips. And then we normally use HPLC to analyze or estimate the dose on those glass cover slips, or 3 of them, to find out if the amount of analyte is as we want it to be, and also especially if you're going to compare some formulations, then you want to have the same amount of analyte of different formulations, and then you need to do some kind of dose estimation before the experiments. But then you still have like 6 glass cover slips left, so you have really enough to do dissolution experiments. Usually we use 3 of the glass cover slips to perform the experiments in triplicates. And here is the setup that I will show you in the lab. So the dissolution part on the microscope, we have the pumps here with the perfusate, the fraction collector, and the computer that controls or steers everything. And as I said, later on, those samples will be analyzed by using LC-MS, and by that you will achieve this type of concentration profiles. And I will also, before taking you to the lab, I will just briefly present one recent study that we have performed in collaboration with Hovion in Portugal. And here we looked at different ways to formulate substances. So first, and compared this commercial available Flixotide with 2 different formulated or different micronization technologies. So we have a jet milled and wet polished micronization of fluticasone propionate. And here we see dissolve it profiles, concentration profiles over time for those 2 FPs that are jet milled or wet polished compared to the commercial available Flixotide. And we could see that at this time point they are differs from the commercial available Flixotide. But then those have been formulated, so we have blends that contains then lactose and excipients, and we have the jet milled form that is formulated, and you have the wet polished formulation that also have been or has been formulated. And then you could definitely see how this jet milled blend really correlates with the profile, the dissolution absorption profile of Flixotide. So this could really be one way to check into your different micronization or formulations to compare an original product with different test candidates that you have. And I will talk a little bit more about that in the lab. So then I will go to the labs. See you soon again.
Thank you, Maria. Excellent. And I just wanted to mention there are many other articles with dissolved data, of course, that we have published. But this one is very special to me because it's very recent, having been published, I think, maybe 2 months back. And then it's just this, like, comparison showing the possibility of dissolving, of discriminating across formulations or micronization methods. So we thought it was a great match for the discussion today on generics drugs. Then, of course, also Mikael just hinted very quickly at the possibilities what PreciseInhale as an aerosol generator can offer in vivo. And if there is interest, we will be happy to engage the audience again in another webinar, or maybe do more meetings, one-on-one dedicated meetings, and just get in touch with Apex Inhalation, and we will organize it. So I think now in these COVID times, these video meetings, everybody got more used, but we thought they work in an excellent way, and my colleagues could manage even installations with kind of live meetings. So now I'm stopping the presentation and closing the video, and we move over to the lab. Please, Maria.
Yes, thank you. Now I'm here again from the lab this time. So here we have the setup of Dissolvit. We have here where the dissolution takes place on the microscope. We have the pumps, the fraction collector, the computer that steers everything, and the control box that has the electricity within it, and also, maybe the microscope if I didn't mention that. So I will take you through a typical experiment here. What we start with is this glass cover slip that we have received from the holding chamber where the aerosol has been deposited, as Beatrice showed you within PreciseInhale. And you take this glass cover slip with particles facing upwards, putting it in place on the dissolution assay. I know that it's hard to see, but I will explain more around it. And this dissolution assay is heated to 37 degrees to simulate the body temperature. So now we have the glass cover slip with the particles facing upwards. And then we have the dissolution chamber, where we will apply our mucus simulant, as I talked about before. And the mucus simulant will be applied on this side, and the thickness of the mucus simulant is defined by this green ring, and it will be 50 micrometer thick. And here you also see inlet and outlet of the dissolution chamber. So the mucus is applied on this underside, and on the membrane, and on the other side of the membrane, the perfusate is pumped in and out. So now I will put this in place in its little holder. So we have a little holder here. And this holder will also be heated to 37 degrees when we place it at the spot of dissolution. It means that everything will be body temperature. Secure this with 2 screws. And as I said, the tubes here are inlet and outlet of the perfusate. And the particles, as you remember, pointing upwards on the glass cover slip. The mucus is applied on this membrane, which will be put like this. But when I put it in place, you still will not have contact in between the particles and the mucus implant, because you have a slope here in this dissolution chamber holder, and that slope will make it possible due to the construction of the dissolution assay that the particles is not in contact with the mucus implant until we start the experiment from the computer. So now I will put it in place here. So here it is now put in place, but the experiment is still not started. So what you also prepare on the computer is your sampling schedule, and then you are ready to start your experiment by just pressing a button, start. And what happens then is that the particles get in contact with the mucus implant, and the particles are starting to dissolve. And you could also, in the microscope, you could take photos and videos during the experiment and see how the particles disappear over time. So that is really the dissolution separated from the absorption of particles. The absorption instead is visualized later on with the help of samples that you collect. So the experiment has started, and as I said before, you have those pumps pumping our blood simulant into the system, and that tube is actually within the system here, so the perfusate is then warmed up to 37 degrees. So everything is at body temperature. Then it comes, when it has passed through the dissolution chamber on top of the membrane, where the particles that have been dissolved into the mucus could be absorbed into the perfusate. The perfusate is further transported here to the fraction collector. And as I said, this could be programmed on beforehand. You have this rack with microtubes, so you could use up to 64 samples per experiment, which is really a lot. And those microtubes you could collect from 35 microliters up to 1.8 milliliter, and that corresponds to, depending on the default flow rate, that is 0.42 milliliter per minute, then that will give 35 microliters up to some milliliter of sample. So within this, the samples are collected during the experimental time. So when the sampling happens, you have this thing going, the tube going up and down within those tubes, and then it's moving automatically. And the drain volume that you're not sampling is just going to the drain flask here. And then when you have finished your experiment, you have your perfusate samples here, so you just cap them and put them into the freezer until they are able to be analyzed. You also save this dissolution chamber. As I said before, the mucus simulant got in contact with the particles on the glass cover slip, and this mucus simulant is a bit sticky, so the glass cover slip is normally adhered then to the dissolution chamber via the mucus. So you will get out like a package here with undissolved and unabsorbed substance. So actually there could be undissolved particles left on the glass cover slip, depending on the properties of the tested formulation, and also some unabsorbed substance within the mucus simulant. So this one you also save into a tube, and this will also be extracted and analyzed by LC-MS analyzers. So that is what happens next. You either have it in-house or you send it to a third party to analyze those samples. And what you get out then is the concentration values for all the samples, both the percocet samples and the system wash or the residue sample left in the system after the experiment. And what can you then do with those type of results? To the left here, you could see how fluticasone propionate and budesonide have been tested in Dissolvit. Concentration plotted over time, and there is also different time scales. So here it's up to 8 hours, here only 1 hour. And you could actually run experiments up to 8 hours and as short as you want. And those two could then be, as I said, you will get profiles with Cmax and Tmax, and those could directly then be compared to clinical profiles, for example. So actually, those graphs to the right are graphs found in the literature, published data on fluticasone and budesonide, how it looks. And as you could see, the appearance of the graph is very similar. Another type of data you could summarize from Dissolvit is if you work with generics. Graph you just saw briefly in the presentation, but here Dissolvit is excellent to use if you have a reference product and some generic candidates that you want to compare to the originator product, because then you can run all the formulations within Dissolvit, plot the curves, and compare the originator product dissolution absorption curve to your test formulations. And from that, you could draw conclusions then on what candidates should you continue with, take further into your development. That is briefly a description of Dissolvit. And I give the word back to the seminar room. Thank you very much.
So, thank you, Maria. I can introduce Per here, Per Gärde, our founder and the Chief Scientific Officer of the company. Pleased to meet you. Yes. And I think we are ready for questions and
for Q&A. Paolo here. Now Maria will join us very shortly from the lab. And yes, definitely, Mikael, we are ready for the Q&A session. So we are still sharing our screen. Kumar, please tell us if we need to do anything or at this point.
Hi, sir. Generally, if you have any questions or clarification regarding this presentation, you can put it on Q&A tab showing on left side of your screen in your control panel. You can ask for any presentation or any PPT shown by Paulo sir or Michael sir or Maria.
We got one question for Maria, but maybe Per, we got one question on Dissolvit. And there is the question how the Dissolvit data correlate with in vivo data. Since it's generated in vitro, maybe we'll let Maria and Per answer this one. Maybe Maria, you want to start? I think we have to. Yeah.
Yeah, hello again. From the seminar room again. Yeah, so you were talking about the correlation of in vitro and in vivo data. So did you have those slides, Paolo, that you could
No, I mean, that should actually not.
Yeah.
Not to reply, maybe.
Yeah, but we have done some studies, especially comparing the dissolved data with IPL, which is the isolated perfused rat lung, which gives you specific PK data from lung. And we there could see that it correlates very well, both for fast dissolving substances and slowly dissolving substances. Just that difference that, for example, or for dissolve it, we have, of course, as every other in vitro method, a thicker barrier compared to the IPL. How do you want to add something?
Yes, I mean, like Maria is saying, in the isolated perfused lung, then we have the advantage of being able to measure the blood concentration exiting the lungs. Because if you go to all clinical data, there blood sampling takes place on the blood returning to the lungs. So there you're a little bit farther away for getting a strict correlation between an in vitro dissolution and what you actually measure in the venous blood. The IPL gives the great advantage that there you actually sample the arterial blood exiting the lungs. But even so, there are pretty good correlation also with what we have seen with the clinical data. Of course, our goal in the near future is to get correlation for exactly the same formulations. I mean, we have compared for the same substance, but in different formulations and found reasonably good correlation. But our goal is to actually run this correlation study on exactly the same aerosol, which we hope will sort of challenge the models further, both our in vitro models and other in vitro models. So,
and we can also add that we have performed some studies for different customers that has tested their products in clinical studies, and we have tested it in Dissolve It. But of course, we are not able to publish those data, but we could say as much as that it seems to correlate very well according to those people who has tested it both in Dissolve It and clinical.
Thank you.
Here is one more question. I think is asked by Remy Rosary. It's asking, could PreciseInhale be used as QC to ensure batch-to-batch repeatability? For example, spray dried powder.
Could PreciseInhale be used as QC for quality control to ensure batch-to-batch repeatability? For example, spray dried powder. I mean, we can comment maybe on PreciseInhale and Dissolve It as well. So I mean, the whole system together. Yeah.
No, but I think this is an excellent approach to generate dissolved data from different batches, so that you can have straight comparison between them. So
probably one comment is that the need comparing batch to batch would be more in the, let's say, details level, so no major variations. We're not talking PK profiles across different formulations or different APIs. So we think Dissolve It would have the capacity to discriminate across batch to batch. Now, I think I didn't mention, we have a discussion ongoing with the agency. Our objective is really to get Dissolve It to be a reference test method. But, it's an ongoing discussion, but definitely will be one of the possible applications.
yes, definitely. Although it's because it delivers more in vivo-like data. So perhaps you could pick up batch to batch variability even on simple dissolution methods. But with the PreciseInhale, you will get it put into a more in vivo-like context on how it varies. I mean, we can check on parameters like Cmax, Tmax, and things like that also, in addition to overall area under the curve, which perhaps you can do with any method. So yes, we think definitely QC as well, and with greater certainty to the data.
Thank you, Per. Now we got a third question, and this is asking about the comparison of Dissolvi to the other dissolution methods. We had a bit of a glimpse of it in Maria's presentation, but maybe we can start with Per this time and then go over to Maria, a little bit of a kind of, recap.
Yes.
What were you thinking when you designed Dissolvi,
Yeah.
Per?
No, I guess the idea we strive for was to actually deliver data that is more in vivo like, so to speak, that we do run the single pass type perfusate, which gives more clinical like systemic, so to speak, blood curves compared to the typical cumulative curves you get in a standard in vitro test. So yes, that's definitely one advantage with it. It lies within, so you will have more in vivo like mechanisms involved in the dissolution and the absorption process in the in vitro system and as well in vivo.
You want to comment further, Maria? Anything else you want to add?
Is there any more questions? If anyone have questions or clarification regarding the session, you can write it here only.
So as I mentioned, yeah, sorry.
I think here in admin, I can get one. I'm just seeing one question here, sir.
Please.
So it's asking, can we determine the Pricklet number for dry powder inhalation systems?
Can you please repeat that, Kumar?
He's asking, like, can we determine the Pricklet number for dry powder inhalation systems?
The preclinical number.
Yeah, PE in bracket
Elaborate
glycerin PE.
Is it possible to elaborate the number?
Wait. I think so. Yeah. Are you able to see, sir? Can you please go to your Q&A tab?
We don't see any more questions on the Q&A.
It's written, yeah.
Here.
This one, sir.
The Pecklet number, Pecklet. Here. You see, Pecklet number. Is it the parameter for dry powder inhalation system? Pecklet is it the physical parameter?
Yeah, that's one of those dimensionless numbers. Now you're really diving into the details here. See, are we? Do you mean defined on the aerosol side, or once it's on the deposition side, during the dissolution phase or during the aerosol phase? Because I think you can define it on in both environment as well.
Because we could maybe, this is coming from Mr. or Mrs. Garima. Maybe we can follow up on this one offline. It's a very specific question.
Yeah, I think we need to get back with.
Hope Garima is satisfied with the answer. Hope Remy, Rosary, you satisfied with the answer by our panelist. If you have any more clarification, you can write to me at raju@PreciseInhalation.com. We will revert to your query, and if you have any clarification regarding whole presentation, you can write to me. We'll forward your question with Paulus and whole team of Inhalation Sciences, and definitely you will get the right answer and clarification from Inhalation teams.
Absolutely.
Yeah.
Let's do like that.
This session, if you missed the session, if you missed today's session, this session is available on YouTube for tonight till 12 o'clock. You can go and watch Even if you have any clarification, then you can comment it with your clarification. We'll pass your questions and clarification with Inhalation team.
Absolutely, we
Yeah.
reply all comments and questions. And again, this is just the first kind of webinar we organized today together with Microbios and Apex Inhalation, but we could follow up with other webinars. And actually, if you have interest in specific subject on inhalation pharmacokinetics for inhaled drug products, please feel free to comment on the YouTube video or send an email to Apex Inhalation. Check our website, so we will follow up and we can set up a webinar if there is a specific request or on any kind of specific area that we can cover. Definitely will be our pleasure. Any other question coming up? I guess not.
Yeah. Let's wait for another 2-3 minutes more, sir.
Please. Yeah, absolutely.
Yes.
Don't be
Those
shy.
attendees are still here.
We have the opportunity of having here with us Per, so we should just take advantage of this.
Sir, one question from my side. Is there any link on Inhalation Science website where attendees can go there and download any application note after filling databases and all? After filling their contact address and position, they can download the application note on that power of precise PK data in generic drug development. Is there any website link? If is there any website link, and request you to kindly paste it here in Q&A tabs and broadcast here only. Attendees, you can go there and fill your database and fill your contact details and position, and you can download your application notes. Is there any possibility on your website, sir?
Not as of today, but we have a list of published articles on our website, and but it's definitely, we want to make available this presentation somehow, so we just have to discuss maybe the best way to do it. Maybe just we can do an email, a follow-up with the registrants, and follow up with the Apex Inhalation or through our contact email also.
After this session, you guys will get the thank you mail from our end, and you can get the link of Inhalation Science website and Apex Inhalation website, and you can go there and fill your contact details, and you will definitely get the relevant information from their platforms.
The contact
even from
email
Inhalation Sciences and Apex Inhalation website.
Exactly. There is a contact email address on our website, and definitely through that you get through to us directly. And just let us know what your specific interest is, and we will be happy to share and set up more specific meetings. Yeah.
Meanwhile, we wait for questions. I can just clarify that we do provide contract research services and tech transfer, so not only services. And we have installed this instrument worldwide in Asia, Europe, and US.
Yeah, let's see if there is any more questions coming up. I hope the kind of level of the presentation was a good level of detail, which was a compromise between PowerPoint and live demos. We didn't want to put the audience to sleep on a Friday with 1 hour of straight PowerPoint, so I thought it's excellent to kind of get live demonstrations from Maria and Beatrice and see in reality. That is our lab, so that's where the research services would be conducted. So you could see really a little bit of our home here in Stockholm, in Sweden.
Sir, here is, sorry to interrupt you, here is one question.
Yeah.
You're able to see, sir? He just put it on Q&A tab only by Mr. Sunil.
Yeah.
He's asking would you able to see, sir?
Yeah, how does one calculate fine particle fraction by the dry powder inhalation formulation? Fine particle fraction from the formulation.
Yeah, that typically it's defined as the weight fraction of particles smaller than 5 micrometers aerodynamic diameter. And that we do kind of during the characterization of the powder, depending on whether you are running it as a just a powder sample, or whether you're picking the aerosol off an inhaler. But the definition is kind of the same.